Welcome to the Philip J. Currie Dinosaur Museum Virtual Speaker Series, where we have paleontology guests call in for a presentation on science, fossils, and dinosaurs, and where the public will be able to ask questions live on YouTube. First off, we want to acknowledge that we are on the homeland of the many diverse First Nations and Métis people, whose ancestors have walked this land since time immemorial. We are grateful to work, live, and learn on the traditional territory of Treaty 8. Today, Filippo Bertozzo is joining us from northern Italy to present Pathologies Out of Time, Fossilized Lesions and Diseases in Hadrosaurs and Their Significance. Filippo is an Italian paleontologist with a Master of Science. His fascination with the world of dinosaurs began in the early 1990s, thanks to watching the famous Italian documentary Planet of Dinosaurs produced by Piero Angela in 1993. He received a Bachelor of Science in the Natural Sciences at the University of Bologna after studying the skeleton of the ornithopod Tyrannosaurus nigeriensis, exhibited at the Museum of Natural Science in Venice. Then, during his master's in paleobiology at the University of Bonn in Germany, he defended his thesis about paleohistological analysis of pneumatic bones in Saurischian dinosaurs. Afterward, Filippo spent a research year at the Free University of Brussels, where he won a Marie Sklodowska-Curie PhD fellowship at Queen's University of Belfast in Northern Ireland to study the paleopathologies in ornithopod dinosaurs. His research activities focused on paleobiological reconstructions of plant-eating dinosaurs, especially derived ornithopods such as iguanodontians and hadrosaurids. Filippo has done fieldwork in Portugal, Spain, UK, United States, Canada, and Far Eastern Russia. Thank you for joining us today, Filippo. With that, I will let you take it away.
Okay, thank you very much, Carla, and welcome everybody to my talk. Today I'm very happy and excited to present to you a bit of my work, of my PhD work, about pathologies, or better, paleopathologies in hadrosaurian dinosaurs. We will see something about the paleopathology in general, but then we will focus more on these beautiful and exciting dinosaurs, which are extremely abundant in Alberta, and this is also one of the reasons why I wanted to study these animals, because they are extremely beautiful, and the high amount of specimens is perfect if you want to study interesting topics such as pathologies. But first things first, here, who am I? So you already covered a lot, Kayla, about myself, but because I finished like a month ago, I was a PhD student at Queen's University Belfast with my main supervisor, Professor Eileen Murphy, my co-supervisor, Alastair Raffle, and my external supervisor, Dr. Pascal Godefroy from the Royal Belgian Institute of Natural Science in Brussels. I spent the last 3 years working on pathologies in ornithopods, especially in hadrosaurs. I had the fortune to collaborate with amazing scientists and also artists. For example, here on the left, you can see myself studying a beautiful and extremely important hadrosaur tail from North Dakota. And here I am exactly in North Dakota with Professor Denver Fowler and Liz Friedman Fowler. And we were checking the exciting things about this tail. And this tail will say something about this animal, these dinosaurs, especially for the pathologists, but things are on the work. I think you have to wait a bit about it, but believe me, the waiting will be extremely rewarded because there are exciting things going on here. So I started my, let's call it, career in paleopathology by describing a Canadian hadrosaur, actually, in 2017 with a team of Italian paleontologists, Cristiano Del Sasso, Matteo Fabbri, Fabio Manucci, and Simone Maganuco. We redescribed a, let's say, nearly complete hadrosaur Agriposaur from Alberta, from the Dinosaur Park formation, Dinosaur Provincial Park, that arrived in Italy, if I remember correctly, it was 1960 something, 59, 60. And it was amazing because at that time I didn't know I was going to study pathologies, but this dinosaur already presented a few pathologies, a few paleopathologies, which I'm gonna show you later. And recently, we also published an important paper about probably the most beautiful dinosaur in the world, in my honest opinion, the Parasaurolophus, again, an hadrosaur from the Dinosaur Provincial Park, which is very important from a paleopathological point of view. I'm gonna explain this quite a bit later. For this, I need to thank all my colleagues, especially Darren Tanky, the senior preparator at the Royal Tyrrell Museum there in Alberta, who is I like to name it my pathological mentor, since he taught me so much about. So what about paleopathology? Paleopathology is an early old and young discipline in paleontology. What does it mean? Paleopathology is one of the oldest discipline within biology, archaeology, and paleontology, because you need to think that the study of fossilized lesion and diseases has started from the beginning of, for example, archaeology. People were finding mummies and Vikings and medieval settlements with a lot of people with a lot of problems in their bones, infection, tumors, arthritis, and yada, yada, yada. So the study of these maladies started early. However, paleopathology for dinosaurs is extremely new, because we have some things in the past, but the new way to describe pathologies with amazing techniques like histology, CT scan, even synchrotron is pretty recent. So this is like a growing field in paleontology, in dinosaur or even vertebrate paleontology, that there is so much to work on. You have no idea. So paleopathology, I'm pretty sure it will become something very important in a few years. But everything started early, as I said. In 1923, Roy Moody published this important and historical book that is Paleopathology, an introduction to the study of ancient evidences of diseases. This was the first attempt to kind of collect everything that was known at that time about pathologies, both from archaeology and paleontology. In fact, when you go through it, you can see things about humans and even invertebrates. I'm going to show you something about in the next slide, actually. Speaking about dinosaur paleopathology, there was a gap, a virtual gap of 40-50 years in publication history between Moody and the important and new publications of paleopathology. And everything started thanks to a bunch of people, for example, Darren Tanky and Bruce Rothschild, who started early in the 80s to think more about dinosaur and even extinct reptiles and birds and mammals and amphibians pathologies in the fossil record. And one very important paper is Tanky and Rothschild 2002, where they recollect what was known about dinosaur paleopathology until 2002, starting from the first dinosaur discoveries in the late 19th century. And then there is another paper that was important also for my thesis, and is also an important paper to understand what paleopathology can say us, what can tell us. That is the osteological paradox of Wood and colleagues from 1992. We can speak for another half an hour about the osteological paradox, which I'm not gonna do because people here might be more interested about dinosaurs. But basically, the osteological paradox gives guidelines to understand the impact of pathologies in the fossil record. So I'm just gonna throw the question there without answering too much. If we find a skeleton with a lot of pathologies, paleopathologies, a broken ulna, infected vertebra, a tumor in the skull, and a skeleton without any pathology, let's take a step back. The moment before the animal died, which one was the most diseased specimen? The one without paleopathology or the one with paleopathologies? The answer is not that easy, and I'm gonna leave it there. If you want to, if you want to hear more about this, you can read the paper and see discussion on internet. But it's an extremely compelling topic that served a lot in my PhD, actually. A few examples from Moody, as I said before. So you can see here how they were portrayed back in the 20s. This is a femur of a cave bear with what was thought to be a fracture in the middle of the shaft. These are on the left, the first two, the one with B and the left one. These are neurospines from Dimetrodon and Edaphosaur, I think, with gnarly looking jagged bone with fracture and perhaps infection. And the one on the right is left radius of a Dimetrodon with a possible fracture. And the callus is the swelling of the bone during the healing process of a fracture or a trauma. And this is probably one of the most famous examples in dinosaur paleopathology. These are actually two fused vertebra of Diplodocus DP, the Diplodocus carnegie, the one with several replica around the world, one in London, one in Bologna, and in other cities. In the tail, there are two pairs of caudal vertebra with fused centra with what was described later by Rothschild as diffuse idiopathic skeletal hyperostosis. Even there, we don't know if it's a physiological problem or a pathological condition. But in pathology, sometimes it's very difficult to arrive to a specific diagnosis because we have only bones, so we don't have soft tissues, we don't have the genetics, we don't have molecular studies. So you can already start to see the problems in pathology based only on the fossils, on the bones. We have some problem in the diagnosis. Sometimes we are lucky to have clear evidence of what was the problem. For example, a fracture is pretty visible. Other conditions, little more difficult. Probably the most famous example of paleopathology for the audience, for the general audience, is Bigal, the Allosaurus, which I think now has been redescribed as Allosaurus gimazzeni. That was the protagonist of the BBC Walking with Dinosaurs special, The Ballad of Bigal. And I'm pretty sure that has been the first ever documentary dedicated to a pathological specimen. So it has an important position in the history of the discipline. We really need to give a lot of credits to Bigal for several reasons. Bigal is a beautiful specimen that I was lucky to see in 2019 at the MOR, the Rocky Museum in Montana. This is the skull. And these are pictures that I did in the museum. This is the famous broken phalanx of the third digit, the one that in the documentary was depicted to be caused by a fallen on the ground after running. I think they were Dinosaurs. Yeah, I think so, if I remember correctly. And this is instead from the paper that was published by Hannah in 2002, where she went through all the pathologies of the specimen. And here, for example, you can see a callus on the chevron on the right, or a broken rib in the center. In my PhD, together with the osteological paradox, I used, and I wanted to share, if you are interested, 4 papers that really inspire me to define the methodology and the requirements of how to study dinosaur pathology and how to use them to describe behavior, ecology, and other topics about dinosaur. So the first one is about Stilson, is from Stilson, by Stilson, excuse me, Stilson and colleagues in 2016, about the osteopathology of Rhinocerotidae from the Eocene up to now. They went through a lot of bones, a lot of fossils, especially in the limbs of this rhinoceros, to describe the presence, especially of arthritis in these animals, to see how the evolution of the body, so from small animals to large animals, influenced the impact on osteoarthritis in the limb elements. A pretty recent one by Judith Pardo-Perez and colleagues. Judith studied the distribution of pathology in ichthyosaur, the marine reptile, to subdivide pathologies in the body shape of ichthyosaur to see if there is any information about, I don't know, the tail that is being used more for some morphotypes, or other morphotypes were biting each other more fiercely than others. And it was funny because I was in Brussels in 2019 for the European Association of Vertebrate Paleontology meeting to speak about my PhD, and I saw Judith coming out with this methodology that is exactly the same methodology that I also thought to use for hadrosaurs. We will see in a second. And I was like, wow, we independently arrived to the same methodology. That means I'm not crazy to think that morphotype analysis can be good for pathology. So like, hey, nice to see that. Another one is by Molnar, 2001. Molnar did a survey, a statistical survey of pathologies in theropods, the carnivorous dinosaurs, mostly carnivorous dinosaurs, from the literature, and to see again in which part of the body, the head, the neck, the tail, the trunk, the limbs were most afflicted. And the last one by Sivier et al. 2020, she did an amazing job on an Edmontosaur bone bed. She described a lot of pathologies from a single monodominant bed of Edmontosaurus to see in which part of the body the pathologies were mostly focused. And you can see here a pattern. Osteopathology rhinoceros to see in which limbs, Pardo-Perez with the body position, body location of pathologies in ichthyosaur, statistical review in theropods, and from a monodominant bone bed of hadrosaurs to see in which part of the body we see more pathology. So I collect all of these ideas to see, okay, what we will see if we apply this to the entire clade of ornithopods, especially hadrosaurs. So from this, we need to start to think what can we see in a bone in terms of pathology, of course. So let's take a bone, a femur. We can find trauma or lesions. We can find infection, tumor. Tumors are actually a very interesting topic to focus on. Developmental problems, scoliosis, congenital problems, and others. Arthritis, and one specific developmental problem that I decided to consider it apart is osteochondrosis. Osteochondrosis is extremely abundant in hadrosaurs. So abundant that I wanted to give it a proper box, a proper composition, a proper definition in respect of general developmental problems. The thing is that most of these conditions are connected to each other most of the time. You can find a trauma that led to an infection, or an infection weakened the bone that was later broken because it was infected. Or the osteochondrosis might be influenced by trauma in the limbs, for example. Or repetitive trauma can cause tumors. Or infection can lead to arthritis and vice versa, such as in septic arthritis. Arthritis can be connected to developmental, and all of them together can be connected. So to define a diagnosis in a bone, in the bone from the fossil record, is difficult because you really need to know what a normal bone looked like, and also, okay, I see an infection. Do I see any other things about any other condition or problems here? And the things are extremely complicated because you also need to think about that we know a lot about human medicine, but of course dinosaurs are not humans, so we need to go to see crocodiles and birds, but the medical knowledge of these two clades are not as extensive as in mammals, for example. Think about dog, cats, all the veterinary studies focused on them, horses and sheep and goats and cows for alimentary industries. So we really have to think how to define our pathologies, our conditions, based on what we know in living animals. Unfortunately, we don't have any living dinosaur except the birds, but even there, from a Parasaurolophus to a bird, the jump is extremely long. So even there is, yeah, it's tricky to give such connection between the points. So for my PhD, I saw that I have mainly three classes of animals. The first one is the basal ornithopods. So small, agile animals, mostly bipedal, with some problems, not so many. Then we have the iguanodontians, so with iguanodon, Oranosaurus, Altyrhinus, and other species. Here is a bone from the museum in Brussels. And then we have the hadrosaurs, the hadrosaurids, with a lot of problem. Hadrosaurs really suffered a lot in their time. How to connect all of this? If I want to compare and study the influence of pathologies in the clade, the frequency, the differences in body position, what should I do? So let's take an entire clade, the entire clade of Ornithopod. This is from my paper in 2017, where we redescribe the paratype of Oranosaurus, iguanodontian from Africa. And with some degree of freedoms, we can condensate, we can focus three areas of the tree into three, let's call them morphotypes. Basal ornithopods, such as Drysaur, Ipsilophodon, even though some people don't consider Ipsilophodon as an ornithopod, but I decided to consider it as an ornithopod. Zalmoxes, Tenontosaur, although Tenontosaur is very strange because it's also quadrupedal, so it's kind of tricky. Then we have the intermediate portion of the tree with Iguanodon, Mantellisaurus, Lurdosaurus, Ipsilospinus, Oranosaur, and a lot of other names. And then the hadrosaurs, Corythosaur, Parasaurolophus, Gripposaur, Velafrons, Ipacrosaur, Edmontosaur, you name it. So we can define three morphotypes in the evolution of ornithopods. And since these three morphotypes are phylogenetically related, basal, intermediate, and derived, we can compare each other because they have the same body areas. Here I subdivide the morphotypes into 16 body areas: head, neck, dorsal vertebra, sacral vertebra, forelimbs, hindlimbs, hand, foot, etc. So that we can compare each other. If we find, I don't know, 25 pathologies in the skull of Hadrosaur, the third morphotype, and we find only 2 pathologies in the skull of morphotype B, Iguanodon, what this information tells us? about the impact of pathologies on the animal. So the base of this methodology that I'm still trying to make it better with statistical values and stuff is that this can serve as basket for pathological information. Here is a problem that I also encounter in my thesis, especially hadrosaurs. You go to the Dinosaur Provincial Park, and from the people listening to this call, to this presentation from Alberta, you know very well, you go to the Dinosaur Provincial Park and you find thousands of bones, isolated bones. So you can find phalanx, vertebra, femur, but you will never know if that single bone from, for example, a bone bed of scattered material, you will never know if 2, 3, 4 bones are from the same individual, because these are bone beds from herds of animals. The only way to know that they belong to one single specimen is to find them articulated, but most of the time you find them disarticulated and isolated. So you have a problem that you have, I don't know, 10,000 phalanx, which 3,000 are pathological, but how to define them as, for example, 1, 2, 3, 4, 10, 20, 50 specimens? You cannot. So the value of the basket approach is that you can take all the specimen and count them as a single individual. 50 pests, 50 elements in the pest, 50 pathological elements in the pest counts for 50 individuals. I know it's not correct, but it's the best approximation that I was able to find for these animals, especially in hadrosaurs, where we really have thousands and thousands and thousands of disarticulated and isolated material. So I traveled to North America, Canada, Russia, Europe to survey the main museums, the Tyrrell Museum, for example, or the Toronto Ontario Museum. I went through every single bone I was able to see. I was able to check about hadrosaur, general ornithopods, and I was check them if they were pathological, plotted them in an Excel database from which I did the statistical analysis, which I'm sorry I cannot show you because my thesis is right now under revision from my reviewers. But you know, in a few months, perhaps I will be able to publish it and show exactly what we find. Of course, the beginning of my research was to see what we already know from the literature. And in the literature, we see something very interesting. We see that the main, the highest frequency of pathologies comes from the hadrosaurids. They were more abundant, more common than all the others, so more pathologies. You see there is a smaller value for morphotype B, iguanodons, compared to the basal one. But I think this is from biases, collection bias, study bias. There are things that we need to discuss. So within this 498, we see that most of them are osteochondroses from the hadrosaurids pedals, and a lot of fractures, especially in the vertebra and in the ribs. You can see already here in the differentiation of body parts for entire ornithopod that the main parts are in the tail, with 25 in the proximal, 32 in the middle, 68 in the distal caudas. Then we have quite a few number in the trunk with the ribs and the dorsal vertebra, and again, a lot of bones in the foot, especially from osteochondrosis. So you can see here that there are parts that show higher frequency of pathologies, the tail and the limbs, and parts with lower frequency of pathologies, such as the neck. And you need to start to think about this relation between neck and tail, for example, low pathology value, high pathological values. What can we understand from this? I'm spending quite a few words about this in my thesis, and I'm just gonna send it there to you to think about until my thesis is published, because food for brain, as people say. A few examples of pathologies from morphotype A, just a brief presentation before we go to the hadrosaurs. So this is infective tibia from an Ipsilophodontid from Australia. Beautiful specimen. This is a cast at the Royal Ontario Museum in Toronto. And this is the holotype of Camptosaurus dispar, I think, with a deep and extremely large cavity on the iliac crest. This is an amazing specimen, which I still don't know what this was. Probably an infection, a very gnarly and bad-looking infection that completely changed the structure of the ileum of this poor animal. But, you know, if you have other ideas, you can just write to me. Morphotype B, iguanodons. This is exhibited at the Isle of Wight Museum in the Isle of Wight, south of England. This is a dorsal vertebra from an iguanodon, and you can see here, okay, my mouse works. Here, there is this comma-like overgrowth of bones on the left side of the neurospine. Possibly an overgrowth of bone after a traumatic impact on the back of the animal. And this is a ilium from another iguanodon exhibited at the Natural History Museum of London. And you can see here there is a deep fracture that is paired to the other ilium that is in the collection of the museum. So this animal got a very heavy bump on its butt. So yeah, this animal really, well, yeah, he had quite a few problems in its bottom. And then we go to the other source. Here we have a lot of examples from the literature. This is fused metacarpal from another source that was described by Preto Marquez and Serrano Branas. This is septic arthritis example by Anne et al. This is a recent one. This is a South American hadrosaur described by Cruzado, Caballero, and Powell with this bent neurospine. This is a recent one. This is a series of distal caudal vertebra from Dinosaur Provincial Park with possible tumor Langerhans cell histiocytosis. And then this is a broken, infected rib. Yes, rib from another source. I think this is also from the Dinosaur Provincial Park. So you see, there are a lot of examples in other sources. Other example, this is a beautiful yet bad-looking fused metacarpal from hadrosaur at the Smithsonian with, you can see this huge swelling enlargement overgrowth of pathological bone that is fusing these two bones together. And I so want to do a CT scan of the specimen to see what's happening inside there, because it's very bad looking. Then this is a humerus. Usually humerus has this shape. It doesn't have this triangular overgrowth. It goes here, and then the deltopectoral crest is not as squished and enlarged by like that. But this poor animal really suffered, I think, a huge impact on its forelimb that completely squashed and reshaped the morphology of this bone. And as you have seen this one yesterday, this was yesterday Fossil Friday by the Currie Museum. This is a fused two proximal caudal vertebra from a Lambeosaurine hadrosaur from the Dinosaur Provincial Park with the chevron, which is probably spondylarthropathy, a kind of arthritic-like condition fusing bones in their joint. So a bit more about what I studied before my PhD. This is the Griposaur that I mentioned at the beginning. This is the Griposaur. In this Griposaur that is kept at the museum in Milan, here in Italy, the predentary shows this huge enlargement in the center of the bone that we thought to be osteomyelitis. Maybe this animal chomped on a piece of wood, and the wood pierced through the mouth, and the bacteria started to infect the floor of the mouth and went to eat and remove the bone from the predentary. But I can say now, after all the specimens I've seen so far, that this is pretty unique, and I'm still, yeah, this bone is really difficult. Then we have a dorsal vertebra here in the center with fused and hypertrophied rib, possibly from an impact side to side, maybe with another individual. And then we have two fused caudal, I think middle caudal, in the center of the tail, possibly dish, diffuse idiopathic skeletal hyperostosis. But these still, I mean, I'm not very sure. Maybe it's dish, but again, probably with diagnosis in dinosaur pathology. Then from Siviero et al., I wanted to show you something that is extremely interesting also for my thesis. What did Siviero with her paper? So she studied the pathologies that were found in a bone bed of Edmontosaur. Here you can see some example. This is a vertebral centrum with this overgrowth here. These are all neurospine with overgrowth here and here. This looks very gnarly, perhaps an infection. And here you can see this large callus at the base of the neurospine. So these are common pathologies that you find in Canadian and, well, late Cretaceous hadrosaurids. She plotted all the pathological bones in the large sample she had from the bone bed, and she found that the highest frequency of pathology is in the vertebra, after in the chevron, forelimb, and then the ribs. The fact that we have a lot of pathologies in the vertebra is extremely interesting, because we can go further and see which vertebra suffered more from, let's say, impact, traumas, and other problems. Cervical, 0. Again, think about what I said at the beginning, the difference between the low pathological value in the neck against the high pathological value in the tail. Here you can see that in the bone bed, there are no cervical with pathologies, some in the dorsal, but the main in the caudus, proximal, middle, distal. Siviero further subdivide the tail region in proximal middle and middle distal. And you can see that most of the pathologies are in the center of the tail. Not so much at the beginning, not so much at the end, in the middle. We are working on some hypotheses to describe this trend, because it is a trend. In pathology, you can find trend. You can identify pathological trend, especially when you consider a lot of specimen from one clade. And the fact that hadrosaurs present a lot of pathologies in the tail led some people think that these were because these animals were using the tail for defense. They were living in earth, so they were stomping on each other or bumping on each other like tail to tail, perhaps for intraspecific combats, or even as a mating problem. Perhaps the animals were mounting in the reproduction time, and they were injure each other. These are all open questions that me and my colleagues are trying to describe, and we might have something, but again, sorry to say that, but wait for the paper, because we are working on that, but believe me, there are exciting things on the horizon. So the point of my research, to summarize what I did in my PhD, is that, okay, Siviero and colleagues describe a monodominant bone bed, a a herd, a group of Edmontosaur. But what will happen when we will check the entire Hadrosaurid clade? Do we see the same trend, or do we see different trends? So in other terms, the pathological trends identified in the bone bed of Edmontosaur is reflective of the trends that we see in the general Hadrosaurid evolution, or is specific only for Edmontosaur? I don't know, perhaps a group of Lambeosaur have other pathologies, perhaps in the neck, more in the neck than the tail of Edmontosaur. And this is one of the questions that I wanted to answer in my PhD. Spoiler alert, they are very similar, the Siviero results and mine. So this is the other paper that I wanted to show you what we did with, again, possibly one of the most famous hadrosaur in the world, and my favorite dinosaur, that is the Parasaurolophus vulcari, the holotype, exhibited in Toronto. So this is the original. However, the crest, the skull is kept in the collection. And this animal has a lot of problems, like a lot. They were already identified in 1922 by Parkes, but people never put so much effort to describe them. So they were like, yes, it's pathological, no, it's not pathological, but yes, maybe, I don't know. So when I went there, I was in front of the specimen, and together with the help of the curator, David Evans, we were able to see that this animal really spent a difficult life in the late Cretaceous. So let's start from the head. So we see the head, and we go to see the maxilla, so the bone that brings the upper portion of the teeth. And we see something extremely interesting. A small portion of the maxilla is raised up and swollen to show the alveolar attachment of the teeth. And this is something that has not been recognized in any other hadrosaur. So again, the problem here is, are we looking at a particular anatomical characteristic that is phylogenetically be present in the specimen, or are we seeing a pathological deformation? Once we check the bones with other closely related animals, or even other individuals of the same species, for example, for this, we check other hadrosaurs, we see that this is a pathological modification, not a phylogenetical signal. What is it? We hypothesize for a parodontal disease, but we need in the future to do a CT scan of the skull and see what's happening inside here. There is a specimen in Ottawa that was called the Stephanosaurus that might represent the progression of this problem in hadrosaurs. So the maxilla, the lower margin of the maxilla is enlarged and completely detached from the gum. Let's call it gum, but it's not correct. From the alveolar margin of the teeth, and is projected laterally, like to coming out from the mouth. So it will be interesting to compare under CT scan these two specimens. Then we see the ilium. So the ilium of Parasaurolophus is quite particular. So even Parkes, the paleontologist who described it in 1922, he realized that this part of the ilium, the pubic process, is enlarged ventrally. So it's like going down. But then when we see other, even Parasaurolophus ilium, or at least thought to be Parasaurolophus, we see that this process is normal, because usually in hadrosaurs, this process is not developing so much ventrally. Only in the specimen is going down in such an extremely elongation. So again, is it a phylogenetic characteristic, or is a pathological modification? when I was there, I look behind the bone, and you can see that the ilium is overlapping the pubis and is pressing on the pubis. This is very weird for dinosaurs. Generally, in dinosaur, you have the acetabulum, the acetabular opening where the femur articulates, that is formed by the connection between ilium, ischium, and pubis. But this connection is not like this. It should just lay to each other via cartilage. They don't fuse. Here they fuse, but they don't fuse like on the attachment. They overlap and fuse. This is an abnormal condition. Here, to define the pathology here, we should have look inside the bone to see how the bone reacted. However, the specimen back in 1922, it was attached to the wall and it's impossible to remove the specimen. So it's not possible to do any X-ray, CT, or whatever other internal analysis. So we only need to use our eyes to describe this. And then we go to the most important part of Parasaurolophus, the modification of the neural spine. There are a lot of paleo art reconstruction showing this dinosaur with a saddle on the back. And people were thinking, okay, this is the notch where the animal was, when the animal was looking on the sides, the crest was passing through this deformation, so it wasn't bumping on its back. Not really, because when you go close, you see that on top of the anterior spines, there is this overgrowth, this discoidal overgrowth that is not present in any dinosaur at all. So again, phylogenetical signal or pathological modification. This is pathological. You go there close to it and you analyze it. There are all the information, there are all the characteristics for pathological bones. Then you see in the connection between the vertebra, and you see that there is enlargement of the pre-zygapophysis, post-zygapophysis connection, the articular facets of the vertebra. There is a callus, a small callus. And then below here, in the vertebrae that present these V-like spread neurospines, the ribs are broken with a bony spur here and two calluses here. So something happened to this poor animal. Probably something very hard, very heavy, just strike him, her, we don't know, so hard that basically it broke his back. But it survived, because when you find paleopathology, it means the animal survived. So the body had time to react to the offense and start to heal. The position of this pathology has been extremely important to reconstruct soft tissue in dinosaurs. And you can see here one of the most important points for pathology is that when you study pathology in an extinct animal, you not only study what happened to this animal, but perhaps that pathology can tell you so much more about the body of the animal. So the nuchal ligament is a structure, a soft tissue structure that was that usually connects the back to the head and is well developed, for example, in horses. The position, presence, and extension of the nuchal ligament in dinosaurs, especially hadrosaurs, have been debated for so much. And thanks to my colleague Fabio Manucci, that was able to remember this characteristic and connect the points and say, Filippo, well, you say that the discoidal overgrowth can be diagnosed as myositis ossificans traumatica. So it's a secondary ossification of soft tissue after a trauma. Okay, it's a secondary ossification of soft tissue. The position reminds me of what has been hypothesized to be the position for the nuchal ligament. And I was like, Well, you might have a point, because the myositis might have started from the base of the nuchal ligament. So the position, location, position, and recognition of this pathology in this dinosaur might have given us one of the very first clue to identify the origin position of the nuchal ligament to this dinosaur. Of course, it's not sure, because it's a soft tissue we are not sure about. So we offer a lot of possibilities to reconstruct this dinosaur. Why? The discoidal overgrowth is shared between two spines, the 6 and the 7. So we don't know if the nuchal ligament started on the 6th or on the 7th. Well, we did both. 6 here, 7 here. Then in modern reptiles and birds, archosaurs, the nuchal ligament can either attach to the first, to the axis, to the second cervical vertebra, or the occipital region of the skull. And we don't know where it went in this dinosaur. So we say, okay, let's show all the possibilities. So we did, again, 6 or 7 neurospines to the axis or the skull. Then we also have to define that we don't know how extended were the so-called lamellar part of the nuchal ligament. They could be elongated or more reduced, because there was a paper by Currie Woodruff that saw that even in living artiodactyls, there is great variation between the extension of this lamellar part. So we have some animals with a wide neck, other animals, even closely phylogenetically closely related, with a smaller neck. So again, we wanted to offer another set of morphology for this feature, all starting from pathology. So can paleopathology be used as a tool for soft tissue reconstruction? Apparently, yes. And I want to thank Matthew Dempsey, that is a British PhD student at the University of Liverpool, and also a great paleo artist that made this reconstruction. And he also participated a lot in the discussion to define this nuchal ligament in this dinosaur. And also, what is very interesting is that the nuchal ligament seemed to start on the 6/7 dorsal vertebra. Sorry. Sorry, my screen just died. Wait a sec. I'm sorry, I'm gonna start it again. I had a small problem. So, Kalla, do you see the screen again?
Yes, I can see it fine.
Okay. I'm very sorry, but I had a small problem. So, as I was saying, the position of the nuchal ligament based on the position of the discoidal overgrowth is on the 6/7 dorsal vertebra. And it is interesting that in hadrosaurs, we have a set of so-called ossified tendons on the back, going from the back to the tail. This ossified X-like overlapping firm of tendons that were fixing and hardening the back, and actually they started in the back from the more or less 6 or 7 vertebra. So thanks to the pathology, we might say, okay, if the discoidal overgrowth corresponds to the base of the nuchal ligament, then we can use the beginning of the ossified tendons in other hadrosaurs to define the position of the nuchal ligament in those hadrosaurs that do not present this pathology, because it seems like that the neck area was controlled by the nuchal ligament, and all the rest of the body, from controlled by the ossified tendons. So the pathology was exactly in the middle point between these two different sets of soft tissue. And this is extremely lucky for us. It was unlucky for the poor animal, but thanks for this unlucky event that here we hypothesize to be a vertical impact from possibly a tree, but anyway, a very heavy and big object, because you saw there is the broken dorsal spine and the ribs all in a single line. This is why we opted for the tree, but perhaps it was a rock or another individual. We don't know, of course. And I also want to thank Marzio Mereggia, that is an amazing paleo artist from Italy that gave this amazing reconstruction. So we are very close to the end. These are the future research points from this project. Here you can see some example of what we are speaking about. Thanks to paleopathology, we can describe, perhaps identified behavior between individuals of the same species. The dinosaur that we describe in Milan, the Griposaur, thanks to the pathology in the trunk, so the fused broken ribs in the dorsal vertebra, we hypothesize to be the result of intraspecific competition fighting side to side. So the next step will be to study the influence of pathologies in the biomechanics of the animal. So we have a lot of osteochondrosis, and I'm going to show you in a bit. What does it mean for an animal walking to have osteochondrosis? It's not very well understood yet, even in modern animals. So it's going to be tricky, but interesting. Then the difference between the influence of the habitat. Sometimes some pathologies can be induced by diet, difference in diet. So I was lucky to study an amazing bone bed in Russia from the late Cretaceous. And I'm following the methodology by Siviero, the Montessor bone bed researcher, to arrive to the same result and compare how different were the pathological rates between a group living in North America and one living in Asia. Intraspecific behaviors, already say that, and medullary bone. The medullary bone is tricky. I'm not going to spend too much time because, again, here we can speak for another hour. Medullary bone is a type of tissue present in birds, basically before ovulation, before the production of eggs. Birds like chicken deposit calcium within the long bones that will be used later to form the egg. So people were using this proxy to say something about the sex of the animal, but there might be influence for pathologies. And also, paleopathology is a tool to understand the modern tempo of evolution. Remember, neck, tail, you will think about. So, very quick, the osteochondrosis, it was already been described by Rothschild and Tanke. This is what osteochondrosis looks like in the fossil record. So in the phalanx, you see this depression, very shiny and sharp depression in the phalanx. Very easy to recognize around when you know where and what to search for. And again, there are a lot of them, a lot. But interesting point. It looks like a happy face. Interesting point. When we look at iguanodontian phalanx, there are nothing. And it's not a bias. It's not because we don't have enough phalanx of iguanodons. We have a lot of phalanx of iguanodons, but there are none. Like none. And this is a trend. The fact that there are a lot of osteochondrosis in hadrosaurs and not in iguanodon, it's it's significant. It's significant. There is a reason behind, possibly the locomotion. Perhaps body mass, type of locomotion were different between these two groups of animals. So Ostranchondrosaurus was more prone to be present in hadrosaurs rather than in iguanodons. So something to study in the future. And then the medullary bone, as I said, so you had the chicken. To produce the egg, you deposit calcium within the long bone. So this has been also found in Tenontosaur and in Tyrannosaur. It was quite famous when it was out in 2005, if I recall correctly, from Mari Schweitzer and colleagues. So they were saying, okay, we have an animal with medullary bone. It means that this animal was female, ready to give production, ready to start the production of eggs. But the correlation between medullary bone and sex is it really one-to-one? Because some people, there were some researchers saying that, okay, but medullary bone is also formed when in birds, when there is a pathology. You break a bone, you need calcium to reconstruct the bone. And there is a specimen in my database that is very interesting for this topic. So I'm looking forward to work on that. And evolutionary rates, I'm gonna just show you again this graph that is perfect to describe to describe what I want to say. That can paleopathology trends be used to help understand us how evolution works? I'm just gonna leave it there. So this is the end. I hope you liked this presentation. Sorry again for the technical problem. So this is the end of my PhD. I just finished a few weeks ago and I still have to defend my thesis. But for this, I need to thank a lot of people, especially all the curators and the museum staff of all the museums I've been to in the last, well, 2018, 2019, we know what happened in 2020, so I did nothing in 2020, but until 2019, I was traveling so much and I saw so many museums and there are so many exciting things in the future. And I'm gonna give you, since you arrived so late about this presentation of this crazy guy speaking about suffering dinosaurs, I'm just gonna gift you a small spoiler of the next paper that is from a paleo art piece that I commissioned for this. I'm just gonna leave it there. You don't know what it is. You don't know from whom artist is. Just I leave it there. So you can start theorizing what it's gonna be about. So again, thank you very much for your attention. Thank you very much, Carla, and the Currie Museum staff to inviting me. And I also want to thank so much Darren Tanky from Drameller, because here you can see me and him working on some hadrosaur bones. He has been such an amazing friend, teacher, and especially mentor for paleopathology, and I owe him so much. He taught me so much about. So thank you very much.
Right. Thank you very much, Filippo. That was a very interesting talk. We're now going to move into our question period. So our first question is from David. You group pathologies according to taxonomic groups and body parts. Do you think of checking in the future how they are related to ontogenetic age?
Yeah, so I haven't spent so much about this because again I cannot show things about my thesis for obvious reason. But I had a special look at juvenile material because juveniles there is a great mortality in juveniles. So even there, can we find a lot of pathological pathologies in juveniles or not? Because also, so they were dying young, so they had no time to heal. And also when they were healing very faster compared to adults. So I can say that there are not so many pathologies in younglings, but there are. Not so much as in adults, but there are.
Great. Next question from Cam. You mentioned that there's been a paleopathological analysis on hadrosaurs, theropods, and ichthyosaurs. Has there been much paleopathology work done on ceratopsians?
Yeah, so there is there are two papers about large paleopathological analysis. One is by Farke and Tanke, where they were describing the enigmatic cavities and holes in the frios of several ceratopsians, because we still don't know what those holes are. People think that they might be pathological, but again, there are no living example of similar pathologies in the animal world, so that is difficult. Then there is a large overview of paleopathology in ceratopsian in the book Horn Ceratopsian from 2000, I think 5 or 6, something like that, from the Tyrrell Museum after the symposium about ceratopsian. Ceratopsian are extremely interesting for pathologists because of the fights between horns with holes and infection in the skull, and also some problems in the foot with infection and fractures. At the beginning, my project was going to have involved also the ceratopsian, but because I had only 3 years, I could not check also ceratopsian because I really needed a 6-year PhD if I wanted to include ceratopsian in my project. Perhaps in the future, I really want to start this same project in ceratopsian.
Great. Next question from Sarah. If you examine the percentage of bones that show pathology as opposed to total number, do you still find that hadrosaurs have a much higher occurrence of damage than do the other two morphotypes?
This is the main part. This is the main part of my thesis. So there is a very big statistical problem here. So we can study pathological trends in hadrosaurs, of course, indeed, obviously, because we have thousands of specimens. We have thousands and thousands of non-pathological specimens, and we have thousands of pathological specimens. So we do the proportion and we can see, okay, 0%, 1%, 15%, 20%, 50%. The problem with the other two morphotypes, especially the first morphotype, the basal ornithopods, is that they are quite rare in the fossil record. I mean, yes, we find Ipsilophodon. I also consider the Scalosaur as a basal ornithopod because it was bipedal and blah, blah, blah. Dryosaur and others, but we don't find the large bone beds of hadrosaurs of Montana, Wyoming, Alaska, and Canada. So there you can have the possibility to study the proportion. With scattered material, especially in Europe, of basal ornithopods, I mean, it's like you find, I don't know, 10 pathologies in 20 specimens. You have the percentage of 50%, but that percentage is biased by the total number of specimens. Whereas if you find 10 pathological specimens in 1,000, and for example in the neck, and 600 pathologies in the tail out of 2,000 vertebra in the tail, there you have a significant proportion and a significant correlation between the two. So yeah, hadrosaurs still present a much higher value and frequency of paleopathologies compared to the other two morphotypes, but then we need to really consider if it's valuable to compare directly the three of them.
Okay, thank you. Next question from Paleotufs. Is there any sign of pathologies that were caused by parasites in hadrosaur skeletons and mummies?
There was a paper about parasites in sauropods causing infection, published a few months ago by Aureliano and colleagues from Brazil. And there was a paper about parasitology parasites in coprolites of iguanodon from the Bernissart quarry. But I don't think there has been anything about parasites in Hadrosaurids. I'm not sure. I need to check it, but I don't think I recall having read about that. Something to look on, actually.
Yes, that'd be very interesting to know. Lorenzo would like to know. He says, I found myself thinking about that mummified Edmontosaurus hand. Is it a way to predict how widespread that feature is?
Can you repeat the question?
Sorry, I messed that up a little bit. He says he found himself thinking about that mummified Edmontosaurus hand. Is it a way to predict how widespread that feature is?
I don't understand the question. Widespread. Sorry, I don't get the question.
All right, that's right. We'll move on to the next.
Sorry, Lawrence.
Lawrence, maybe if you could retype the question in the comment section. Right, for now, we'll move on to Lord Trilobite. What's the most gruesome pathology you've seen in ornithopods?
Ooh. Wait, I need to think. The tail that they show with Leeds Friedman Fowler at the beginning. That is a specimen that is visible to whoever is able to go to Dickinson in North Dakota. It's Warwick's duck. This is how it's called. And I have never seen such, I'm sorry, YouTube, I'm sorry, such a fucked up tail like that one. Is a mess. It's crazy to think how this individual was able to survive with such a gnarly looking, infected, traumatized tail. That is absurd. Seriously, absurd.
That poor dinosaur.
And outside Hadrosaurs, there is a pachyrhinosaur from the Royal Tyrrell Museum with, let's say, half a face.
Oh, wow. Amazing that they can survive like that.
Yeah.
Next question from Alexander. What sparked your love for Parasaurolophus? Sorry, I messed that up.
Okay, can you repeat?
What sparked your love for Parasaurolophus? Parasaurolophus. It's a hard word to say.
No, I don't get the second word. What's?
Parasaur.
No, not the name before the question.
Oh, why do you like it? What caused you to like it?
Sorry. Why not? Why not? I mean, it's beautiful. It's elegant, and now it's massive and muscular and impressive and stout, and able to survive a tree on his back. I mean, damn the Tyrannosaur. Parasaurolophus was the real deal in the late Cretaceous.
Very cool. Do you love the different known species equally?
Yeah, I mean, it's dinosaurs. I like dinosaurs. So I prefer herbivores, but I like dinosaurs.
Yeah, fair enough. Next question from Sarah. Now that you're finished your PhD, do you think paleosteopathology will remain your focus?
Nice question. I really would like so. Depends what will happen in my future. Everything depends on what if I get a postdoc and where and on what. But of course, after the PhD, now I have to start to publish everything that I worked on. There are some people that called me for some advices and perhaps collaboration on some pathologies on other animals. And I really, as I said a few minutes before, I really want to continue this project with especially ceratopsian. Especially because ceratopsian and ornithopods, they share the same ancestor. They are part of a family called Ceropoda. And so they are closely related in evolutionary terms. And once you have a pathological overview of hadrosaurs, and then you have a pathological overview of ceratopsian, you can really do comparison between the two because they share a common ancestor. So there might be some interesting thoughts about, for example, the evolution of the immune system, or the differences or analogies of how to react to a trauma, for example, because they are very close in evolutionary terms.
All right, going back to Lorenzo's question, he says he was referring to the hoof-looking hand of Edmontosaurus, and he wants to know if that's just found in Edmontosaurus, or if it's reasonable to assume that other hadrosaurs or ornithopods had it as well.
Okay, now I get the question. I don't know. Because we are still waiting for the official description of that specimen, and like me, all of my colleagues working with hadrosaurs, we are all waiting for that specimen. I know they are working on it, and I can understand it takes time because such an animal deserves a thoughtful study, description, and analysis. I'm pretty sure that when the paper will be out, it will be amazing. So, and when it will be out, perhaps the author will say something about the possible presence of not of this structure in the other source. The point is, there are not so much differences in just the bone morphology of the hand between Edmontosaur and other sources such as Parasaurolophus, Ornithosaurus, Saurolophus, and others. So, from a pure comparative anatomy point of view, I would say that it is feasible to think that other sources had a similar hand, but we wait for the description to say something.
Next question from Carlos. Do you know about any project about theropod pathologies?
There have been some papers about theropods from North America and something from South America. Gutters et al. 2020 from, I think, a bone bed of Majangasaurus, an abelisaurid, I think. Beautiful paper. I don't know if there is any person working on a large pathological review of theropods like I did for others. So this I don't know. So if such person exists and is watching this live, if you want to chat, just, you have all my contacts here. So, hey, maybe we can discuss about something.
Great, thank you. I think that concludes the end of our question period.
Okay.
Thank you very much, Filippo. Really appreciate you joining us.
Thank you for the invitation. It was a pleasure.
For all of you joining in, join us again on Saturday, May 8th at 3 p.m. as Dr. Oksana Vernigora will be presenting the fossil record of fish in Alberta. All right, that's it from our side. Thank you very much again, Filippo. And thank you everybody for joining in.
Bye, everyone.